Hello friends, Ace Botani's next episode. In this episode, we'll be discussing molecular basis of inheritance 8th one. And what all things we'll be discussing in this episode. For that, I will share the screen so it will be convenient for you. I think screen came. So we can proceed. So molecular basis of inheritance 8th one. There'll be as usual an introduction. Then Operon translation is a big area. Then Operon concept and introduction. Next episode, we'll be discussing Operon concept. If time is available, we can proceed to Human Genome Project also. So here, mainly translation and Operon concept introduction. Then as usual, 5 theory question, 5 NEET model question with the key. So we can proceed. As an introduction, translation connected with the protein synthesis, Operon concept introduction. Now translation. See, the word translation means change into another form, translate. Here, in this context, translation means the process of polymerization of amino acid to form a polypeptide. That is what NCERT given in page number 98. It is connected with the bases from DNA, then converting into sequence of mRNA, that is transcription. And after that, it is correspondingly arranging the amino acids, that is translation. Peptide bonds are involved in connecting the amino acid, and peptide bond needs, or formation of peptide bond needs energy. I think up to this, it is clear to all of you. Now, different chemicals and different steps involved in translation. So one by one, I will explain different steps, different chemicals, all this, just like DNA replication. So in this diagram, we can find double-stranded DNA from which single-stranded mRNA will be created by the procedure called transcription that is already over. From this mRNA, that is arranging the amino acids in a sequential manner, that part is called translation. So transcription, translation, finally, protein will be formed. Double-stranded DNA, single-stranded RNA, polypeptide proteins. I think idea clear, first page, so we can proceed to second page. See, in the second page, we can find when we start. So DNA, no doubt, it is the master molecule. So from DNA, message is copied to single-strand RNA through transcription. So from the RNA form, that is from DNA, DNA gives to RNA. From that RNA form, amino acids will be arranged to form protein. That is the sequence, and that is what we are going to study in this episode. And what all things are required, which all chemicals involved. One by one, we can explain. Everything I given, this will be easy for you. One, mRNA. Without that, no message. Second, ribosome. Ribosome, we already know, cell as the basic unit of life, cellular factory for synthesizing proteins, consists of structural RNAs and about 80 different types of protein, 80. Normally, it exists in inactive state in 2 subunits, a large and small. When the small subunit encounters an mRNA, the process of translation of the mRNA to protein begins. So starting with the small subunit, meeting with mRNA. Then third one is transfer RNA. All will be learning further when we proceed, bringing amino acid. Then fourth one, amino acid transfer RNA synthetase enzyme. This is one of the most crucial enzymes. And this enzyme will attach the amino acid to their corresponding transfer RNA, tRNA. And this process is essential for protein synthesis. And this enzyme is playing a central role. And it is otherwise also called center of translation. And that occurs in cytoplasm. I think it is clear because amino acid will be in cytoplasmic pool. So that enzyme occurs in cytoplasm. Now, large number of protein factors, which includes initiation factors, elongation factors, releasing factors, short form IF, EF, RF. Energy in 2 forms, it can be either ATP or GTP. Then seventh one is peptide formation. It is helped by an enzyme called peptidyl transferase. And that is usually located in the ribosome larger subunit, like 50S subunit, etc. This enzyme is a ribozyme. Ribozyme, we already studied RNA-derived enzyme, catalytic RNA. And it is 23 srRNA in bacteria and 28 srRNA in eukaryotes. Now, 8th item is translocation, shifting. which is also known as elongation factor G. In translation, the ribosome moves from codon to codon along mRNA in the 5' to 3' direction. And that statement given in NCERT page number 99. It's an important question. Then reactive sites. See, some sites in connection with the ribosomes is shown. I will next page, I think we can proceed. There I put a diagram. NCERT not mentioned it, but still. So basically, 3 sites we can find. Everything I represent and everything I return there. First is P site, otherwise called D, capital D, peptidyl transfer donor site in the middle of the ribosome. A site, amino acyl site or acceptor site on subunit of ribosome. Then ribosome exit tunnel is a passageway for the elongating polypeptide chain to move through it. So ribosome exit tunnel is a passageway for the elongating polypeptide chain to move through it. Everything marked there, everything written there, so it'll be easy for you. Then comes another site, E site, exit site, is a part of mainly larger subunit to which tRNA lives. A translational unit in mRNA is the sequence of RNA that is flanked, coded by the start codon AUG and the stop codon. And in between, we can find codes for polypeptide, NCERT page number 99. All are NCERT important for exam. And mRNA also has some additional sequences that are not translated and referred as untranslated regions, UTRs. And what is the aim of UTRs? UTRs are present in the diagram. We can find it. It is a labeling diagram. UTRs are present at both 5'N. Here you can find UTR, this gap. Before start codon is AUG. And 3'N after stop codon. This is stop codon, so here. And they are needed for efficient translation process in between coding region. So this will be the range, just like transfer RNA we studied. I think idea clear to all of you. So we can proceed to next page. This is the diagram for protein synthesis in NCERT. That is translation. But previously, different diagrams are there. 2 edition back, another set of diagram that is more clear. Last edition, previous edition, there is another diagram. And this present edition, only this diagram given, but that is sufficient. Everything we can take from this diagram. I'll explain it, how we have to learn this diagram. So main steps in translation. 3 steps will be there. Activation of amino acids, charging of transfer RNA, formation of polypeptide chain. And under this, that is formation of polypeptide chain, 3 headings will be there, subheading. Initiation, elongation, termination. 3 parts will be there. Now we'll start with the activation of amino acid. So proteins means assembly of amino acid. And basic code belongs to DNA. And from there, it is converted into single-strand RNA. From there, the amino acid will be assembled. I think up to this, it is clear to all of you. So we can proceed to next page. So amino acids, that is in cytoplasm, DNA in nucleus. RNA will be coming to the cytoplasm. So amino acid, which all should be arranged or to form polypeptide, that should be activated in the cytoplasm. Then the procedure begins. So enzyme involved is amino acyl transfer RNA synthetase, E, that is enzyme, by which respective amino acid bind with the ATP. That is called activation where ATP is involved. So amino acyl transfer RNA synthetase, that is enzyme, by which respective amino acid binds with the ATP. Equation we can find here, amino acid 1 plus ATP, that is enzyme 1, migration 2 plus required, amino acid 1, AMPE1 complex plus PPI, energy. So that procedure is called activation of amino acid. That is the first step. Here we can find. First step, activation of amino acid. Purpose you understood, I think. Next is second step. We can proceed. Amino acylation, or it is called charging of transfer RNA. Here, that word I have in written. Only charging of transfer RNA, which is technically called amino acylation or charging of transfer RNA. Sometimes amino acylation itself they can ask. It is also called charging of transfer RNA. What is meant by charging, combining, hitting, that way? Activated first amino acid. That is charging of transfer RNA, the activated first amino acid. So first amino acid A1, AMP, E1 complex meet and reacts with the specific tRNA because tRNA has to take it, by which respective amino acid is transferred to respective tRNA. So correspondingly, tRNA will be coming, taking this chemical. It, after attachment, it results in liberation of enzyme and AMP. Energy will be consumed. That is amino acylation of charging of transfer RNA. I think second step is clear amino acylation, charging of tRNA. Third step is formation of polypeptide chain. And this steps involves three sub-steps: initiation, elongation, termination. So first is chain initiation. First step. Initiation requires initiation factor, IF. In prokaryote, it is IF1, IF2, IF3. In eukaryote, it is EIF1A, EIF5B, that way it proceeds. Many are there. Normally, 3 initiation factor in prokaryotes and 9 in eukaryote. That itself is a question. Now, how it proceeds? So, beginning, mRNA binds with the smaller subunits of ribosome. That is how it starts. So, beginning, mRNA binds with the smaller subunit of ribosome, 30S in prokaryote, 40S in eukaryote. So, we will just represent it. I represented here, you can find 30S or 40S plus mRNA, 30S mRNA or 40S mRNA complex. Here you can find. Now, the next event, this mRNA and the first transfer RNA will bind. In prokaryote, it is formylated methionine. And in eukaryote, it is non-formylated methionine attaches with the tRNA. First amino acid. So once again, in prokaryote, it is formylated methionine because no splicing, nothing in prokaryote. In eukaryote, it is non-formylated methionine attached with the transfer RNA. So we can represent, again, an equation I shown there, so it will be convenient for you. 30S mRNA plus tRNA F meth. So it will be prokaryote, formal methionine. GTP energy needed 30S mRNA tRNA F meth prokaryote. While in eukaryote, 40S mRNA plus tRNA, 40S mRNA tRNA in eukaryote. This equation I given this will be convenient for you. After this, next step, larger subunit of ribosome attaches. So larger subunit attaches. 50S in prokaryote, 60S in eukaryote. Ribosome structure you study in cell as the basic unit of life. Now, by this one, initiation getting over, next is chain elongation. So that is chain initiation. Now we can proceed to elongation. This diagram, that is previous NCERT diagram, but it is more clear. That is why I put everything labeled there itself, but I will explain it. So now ribosome is complete. Initiation is over. Complex mRNA, tRNA already formed. Initiation of the peptide synthesis starts at P site. Now ribosome has got originally 3 sites, but we can start with the initial 2 site, that is P site. And amino acyl acceptor site, that is A site. P site has got a name, polypeptide site. A site has got an expansion, amino acyl acceptor site. So after P site, initial attachment, now an amino acyl acceptor site is established next to P site. After that only A site will be established. Because it gives, A site gives next chance for the mRNA codon after initiation codon, that is AUG. So second site came. A new tRNA complex reaches A site because P site is already utilized. And as usual, codon anticodon bonding takes place. Transfer RNA will bring the amino acid and respective code in the mRNA that will be attached in between ribosome. So respective usual codon anticodon bonding takes place for elongation. For this elongation factor, energy in GTP needed. Now 2 amino acids will be closely aligned. This is diagram is small. Next page I expanded it, so you can clearly observe it. Full screen I put it there, but this diagram will not be there in the present NCERT. Here you can find initiation. This is the elongation procedure. So amino acids bringing A site. So this is P site, A site. First amino acid P site, second amino acid A site. I think up to this it is clear. So these 2 amino acids lying very close. You can find it. So when these 2 amino acids lies closely, a peptide bond is formed between these 2 amino acids. Now, which all will be linked in this peptide bond? Peptide bond, you know, it is CONH. So 2 amino acids closely lying, P site amino acid COH group and A site amino acid NH2 group. So A site amino group, P site carboxyl group. See, these 2 charged tRNAs are close so that the formation of peptide bond between them would energetically favor. For this linkage, an enzyme is there that is called peptidyl transferase. that will favor this bond formation. Peptidyl transferase is a type of ribozyme RNA with catalytic activity. 23 srRNA in bacteria and 28 srRNA in eukaryote. And you can find peptide bond is CONH. Now, ribosome, so A site, P site, that is filled. So ribosome able to move through mRNA. It is called translocation. Ribosome will move from codon to codon along the mRNA. Accordingly, respective amino acids are added one by one in A site. Then peptide bond will be formed. Finally, we can find a polypeptide will be resulted. And how this amino acid sequence will be arranged? Sequence of amino acid detected by or dictated by DNA and is represented by mRNA. So sequence of amino acid dictated by DNA, from DNA it comes and is represented by mRNA. Translocation needs an enzyme called translocase. Again, in prokaryote it is EFG. In eukaryote it is EEF2. Now, after the first peptide bond, slipping of the freed tRNA at base site, because linkage is there. So first amino acid brought by transfer at base site, P site, A site amino acid link, automatically P site tRNA will be free. Then the tRNA slips to the A site, exit site. That is how three sites, A site, P site, A site. Now translocation takes place. A site amino acid reaches the P site P site. So now translocation takes place. A site amino acid reaches P site. So now A site is free. So another amino acid reaches into free A site. Again, peptide bond formation between second amino acid and third amino acid. Now that I shown more clearly here. P site, A site is free. Second amino acid came here. Peptide bond is formed between 1 and 2 amino acid. Then translocation takes place. Now both linked amino acids are in P site. Third one will be coming into A site. So diagram clear. This way it continues. This is how it proceeds. Now, next page. Presence of UTRs at 5' end before start codon and 3' end after stop codon to make translation more and more efficient. UTRs, untranslated region. Now the last step in this case, that is called chain termination. It is the last part of translation. Naturally, it has got its own importance. Indication of termination is connected with any of the termination codon UAA, UAG, UGA. No respective tRNA for it. So it is also connected with the releasing factor RF. It is GTP dependent. In prokaryote, it is RF1, RF2, etc. In eukaryote, it is ERF1 that will proceeds. Polypeptide chain will be released from ribosome. After translation, 2 ribosome unit separates and can be reused. And that procedure is called recycling. That is how the protein will be released to ribosome unit separate. Later, if they want, they can join for another reaction. Now, this is the diagram given in NCERT book. So, how we can read this diagram, identify this diagram. So, I will explain it because many people no idea how to deal with. See, this transfer RNA respectively, from there we have to start. That is why I put the line here. From here we have to start. Okay. So, first is here, mRNA GCA, GUU. So, this weight is extending. Okay. So, first when we take GCA, if you check the genetic dictionary, you can find GCA stands for alanine. That idea clear, I think. Now, after that, CCU. From here, CCU from 5' it is UCC. So, it stands for serine. Serine, second one you can find. NCERT diagram. Third is GGG. Here you can find GGG. And GGG stands for glycine. After that again, CUU. It stands for leucine. Now this side. GUU. It stands for asparagine, ASN. Sorry, that is AAU stands for asparagine. GUU stands here, valine. So GUU, valine. AAU, that is ASN, asparagine. Then again, UUC, UAU, that way it continues. This is how we have to read this. We have to identify. It can be a good question for theory and maths. I think idea clear. So first you take these two. Here this one, here this one. Then this one, sorry. Okay. Now this one, after this one, this way it proceeds. I think idea clear to all of you. Yes. It's a very easy diagram. But initially when we observe, we may think it is slightly difficult. But nothing is there. These two we have to take first. Yes. This is how we have to take. C, G, G, C, U, A. This way we have to take. Now it is clear, I think. Okay. Now we can proceed to next page where we can find 5 theory question, 5 NEET model question with a key. But 2 more items you have to discuss there. See, Operon concept introduction. So what is meant by Operon? Who invented, who developed it? What is the purpose? The detailed study next episode. So Operon concept introduction mainly deals with how gene expression is regulated in different levels. Operon is an assembly of genes. All will be discussing in next episode detailed manner. Assembly of genes with a different function. Scientists were Jacob and Monod. Now some protein synthesis inhibitors, mainly antibiotics, chloramphenicol, erythromycin, tetracycline, aminoglycosides. That is regarding this episode. That is molecular basis of inheritance. It is the 8th part. Now 2 more most probably needed. So what all things we learned in this episode? It is starting with an introduction, then translation detailed manner, then Operon concept introduction, then questions. That is how we dealt in this episode. So starting from here. So diagram is there. All explanations are there. It will be easy for you. Here are the chemicals, enzymes involved. Then here we can find ribosome different sites. Then this is NCERT diagram, main steps in translation. After that, procedure started. Then 2 sets of diagram. This is old NCERT. It is expanded here. It will be clear for you, more clear, I think. Then after that, present NCERT diagram is not there. I represented there. How the original procedure, more simple diagram, easy to follow. Then what the NCERT given diagram, how we can read it. That is what I shown here. I think it is clear. This diagram, how we have to start with these 2. Okay, GCA alanine, GUU valine. This way you have to start. Then here AU. Then this one, this one, this one. That way it proceeds. After this, 5 theory question, 5 NEET model question, then Operon concept introduction, then protein synthesis inhibitors, mainly antibiotics. Remaining part will be continuing in 2 episodes. Now, if you like this episode, subscribe all, like it, share it, comment on it. We can meet with another item in the next episode. Hoping that it is clear. Thank you. Take care.